Okay, thank you all for joining us today. This is our kickoff nano lecture for this new academic year. This is part of the NIEHS Nanotechnology Health Implication Research Consortium, which was established in 2016. That includes our center and other 8 research projects. We are all collectively working on assessing the nano-bio interface and assessing potential risks of nanomaterials. And that includes some unique and emerging materials beyond the metal and metal oxides that we've all been studying for years. I couldn't think of anybody better than Tien. Very pleased to have him here. We collaborated in many ways over the years. He's from UCLA. He's an associate professor of medicine, University of California, Los Angeles, as I said, UCLA. He's a known figure in the nanotoxicology domain. He's been working in this area for quite some time. Impressive array of publications and citations. So he knows better than anybody else the nano-bio interface. Of course, you can tell from the title of his presentation, his main focus all these years is assessing the structure-activity relationships of nanomaterials. And he's the best guy to give us a summary of what we know and what we don't know. And, of course, we are not dropping dead from nanomaterials, but I think research over the years helped this community to remove the bad apples and put them aside. And I think that's the major contribution of nanotoxicology research. So we all know that there are good, bad, and ugly chemicals. There would be good, bad, and ugly nanomaterials, and developing methods to screen the huge libraries of emerging materials and putting the ones that we have reasons to believe that they can cause harm aside and develop safer by design approaches. I think that's the way to go. So I won't go through his extensive CV again. He's known in the field. So thank you again, Tien, for taking the time and visiting us, and the floor is all yours.
Thank you, Phil, for your invitation. Also, I'm glad to talk to the postdocs, many of your postdocs this morning. Very exciting work in your lab, so congratulations. So today I'm going to talk about you mentioned, the structure-activity relationship in nanotoxicology. So what we try to do is to understand what properties of the nanomaterials. We use them in the first place, try to know their properties to help us to improve our functionality of the products. On the other hand, they may induce toxicity, and I want to know what properties are driven those toxicity of these nanomaterials. So, as we all know, I don't need to talk too much about this, that currently there are many products having at least one component of containing nanomaterials. So in those products, as we all know, there are some teddy bears. They may have silver nanomaterials to kill all the bacteria that are attached to it. But on the other hand, the children who play with these teddy bears, they may chew on those furs, then they may get exposed to silver nanomaterials. There are other products containing nanomaterials also, like the tennis rackets, carbon nanotubes, carbon fibers are used, lighter weight, but stronger, than the material used before. So as we also know, currently, people are developing nanomedicine. Many of these are, organic materials. They're biodegradable, but people are developing bioimaging agents like iron oxides. Up-conversion nanoparticles contain inorganic components. So these, after they are injected into the body through the systemic delivery. They may accumulate in the liver, to cause some potential effects. So all these we are currently developing for these nano applications that people, on the one hand, they may benefit people. On the other hand, they may generate, potential toxic effects. So as we know that 2016, the President Obama signed a bill. It's a new TSCA bill, the Toxic Substance Control Act reform bill. So this bill demands that all the nanomaterials, all the chemicals should be, ideally be tested before their use to make sure they are safe. So our scientific community has a task on our hands to know how we study the toxicity of the nanomaterials or the chemicals. But to learn from history, we know that the current approach, animal dominant, the gold, standard is animal studies that use a lot of animals. It's highly costly, and the end result is sometimes not translatable to the human, toxicity because of the difference between animals and the human beings. So we have to change this approach, the animal dominant toxicity screening approach. So as we also know that in 2007, the U.S. National Academy of Sciences came up a landmark report that the title is Toxicity Testing in the 21st Century, a Vision and a Strategy. What this report, in essence, is talking about, instead of using an animal-dominant approach, we should use the high-throughput approach that speed up the screening process. Also, we use the cells, bacteria, yeast, those low-level, models to do the screening. And then, when you find any materials that are toxic or they trigger some mechanism toxicity, then you validate in the higher organisms like the mouse. So this process, if you do this way, you can discover the dose-response relationship, the property-activity relationships, as well as quickly identify the toxicity of some engineered nanomaterials. And then, you use this to do in vivo testing. So to give us, some idea of how these materials behave, in vivo. And likely, what will happen if the people are exposed to this. So based on the report, we came up with a scheme on how we study the engineered nanomaterials. So in addition to understand the mechanism of toxicity, we also consider the nanomaterials, different from the chemicals. They have the physical, the structures, the properties. We have to consider this and to develop the quantitative structure-activity relationships and use the cellular assays. We can do a lot, and especially in a high-throughput approach. Then we figure out their, the pathways. Then we prioritize our study in vivo to validate. So this can be done, many times to finally, figure out how these nanomaterials can, induce adverse effects. So To do that approach, a predictive approach from in vitro to in vivo, we have to require some tools. One of the tools is the libraries of nanomaterials. That these libraries containing not only the composition differences, they also contain the different property differences in a library, like the size, surface functionalization, surface charge, surface chemistry, crystal structure, or band gaps. So just to name just a few. So these libraries can help us to identify what property is inducing a toxic effect once you have done all the screening. So then the other tools we require is the toxicological mechanism that are involved in the toxicity in vitro and in vivo. So that the link between the in vitro and the in vivo is on the mechanism of toxicity. So we do screening processes using this mechanism of toxicity. For example, the oxidative stress. It's so far, from air pollution research or other research, chemical research, they are the dominant, mechanism of toxicity. Right? So we have assays to study these. High throughput approach using oxidative stress. Also, there are other mechanisms like lysosomal damage, the NAP3 inflammasomes. Those are the inflammation pathways that the nanomaterial potentially can engage. So using these assays, we can to screen a library of materials, we can quickly identify what the materials are toxic and what the mechanisms are involved. Right? So then, obviously, if it's possible, we should do this in high throughput approach. So, over the years for oxidative stress, we have developed, the microscopic approach, spectroscopy or luminescence, or the multiplex systems. And for zebrafish, we had developed the robotic arm systems to pick and plate the zebrafish embryos to do, different studies. So the high throughput approach obviously can speed up the whole process of our screening. Then, I show here is a example of how we do the, the results of the high throughput screening we use the oxidative stress paradigms using different, parameters like mitochondrial membrane potential, oxidative stress, ROS generation, calcium flux that are involved in the cellular oxidative stress changes, and we screen the 24 metal oxides from this heat map, which covers over 20,000 data points. Each pixel is one, data point. You can clearly see a relatively toxic and relatively non-toxic materials can be differentiated, based on oxidative stress paradigm. Then you can go zoom into the toxic ones to figure out their toxicity mechanisms. So that's basically what our approach are. So obviously, the in vitro screening has to be validated in vivo to see their effects. So for the last slide, I showed some toxic metal oxides, and we can validate that same toxic effects to the lung when we do animal studies, showing on the right side. So these are the tools you need in order to do a predictive, in vitro, in vivo, toxicological screening. So now I gave you an example of how we use it in our recent research, that we also accumulated these metal oxide. Now it expanded to 29 nanoparticles. Then we do the high throughput studies. So these are all the TEMs, you can see. We prepared, we also do extensive physical chemical characterizations, including their size, distribution, their hydrodynamic size, charge, the dispersion, properties. Then we do, to study their effects, in the lung and the liver. So, and the cell system we're focusing on are the macrophages. These belong to the innate immune system that is serves as the first line of defense when the body sees the particles. So they gobble it up and deal with those, exposures. So we want to know their effects on these, innate immune system. As we know, last year, the Nobel Prize, the two winners studied, the cancer immune therapy, immunology. They got a Nobel Prize because they discovered that by using checkpoint inhibitor, they can release the immune system power to eliminate the cancer, to reach outstanding, therapeutic outcome. So what I'm trying to mimic here is to study what the nanomaterials do to the immune system that can affect these functions of the immune system. So what I show here, using the 29 metal oxides, we screened their toxicity profile in the liver macrophage. It has a name, it's called Kupffer cells. These are the cells that situated in the liver as the first line of defense of taking up the nanomedicine, nanoparticles. So from this heat map, you can clearly see three, group of materials, if you will. So we can see that the first one is the first lane is the rare earth materials. That's to us it's a surprise because it's typically considered non-toxic, but it's extremely toxic to the Kupffer cells. The second group of materials are the transition metal oxides. These materials, as I showed in my last heat map, these are transition metal oxides that we know are inducing oxidative stress and kill the cells by a mechanism called apoptosis. And the rest of the materials, rare metal oxides, are non-toxic. So we are fascinated by the surprising finding of these rare earth materials. We try to know what's going on there. So rare earth material give you some background. It's already, although they are called the rare earth, but it's not really rare. It's maybe on par with the other transition metals like copper, oxides. So they are used widely in the different industries like defense, fighter jets, even Mars rover. They contain the neodymium oxide as a permanent magnet. So they are also used in our cell phone screens. So widely used material. Also, people are developing up-conversion nanomaterials use rare earths. These are used for bioimaging purposes. So one recent study in Science reporting that they use those genetic methods that use the upconversion nanoparticles to trigger the activation of neurons, you know. So these upconversion can also use as a bioimaging, you know, for cancer studies. So these are use of the rare earth material. Also, we all know that gadolinium is used as a contrasting agent for MRI imaging in the, you know, clinic. But you know, the patients with kidney disease, they have a systemic fibrosis developed when these gadolinium in contrasting agents are used. That's a potential, you know, adverse effect of these rare earth materials. So what we, as I mentioned, we want to find out what the rare earth is doing to kill those Kupffer cells. What we found, the first thing is we found morphological changes when you treat the Kupffer cells with different metal oxides. The rare earth induce a very unique change, like the cells, it's swelling and bubbling up, sort of like a bubble. But the transition metal, the cobalt and manganese, the cells are not, you know, bubbling up or swelling. The other non-toxic ones, the cells didn't change their morphology. So the transition metal oxides, like the manganese or copper oxide, they are inducing, as I mentioned before, they're inducing a classical apoptosis-like changes. Apoptosis is the cell, you know, after the insults, they become smaller, and the cell then break up into different apoptotic bodies. So that's the what we found for the transition metal oxide induced Kupffer cell toxicity. However, for the, you know, the rare earth, what we found is totally different. That cells, you know, form these bubbles that you showed before, and these cells become larger and form those different they swell and they bubble like an air bubble. So then we try to figure out the detailed mechanism of how this happens. We all know that the cells take the particle up, and for Kupffer cells, those macrophages, the particle are taken up into a cellular organelle. It's called, you know, lysosome. These lysosomes have a low pH, like a 4 to 5. You know, typically their function is to digest food, the proteins, the damaged mitochondria, to remove those debris. And then we want to know what happens if the particles, like the rare earth particles, are taken up into the cell. And we all know that if the lysosomal function is inhibited, like when the cells are lacking of some important lysosomal enzymes, all the storage, the proteins are accumulating this in the lysosomes to cause many diseases, like a heart disease, you know, lung disease, and the brain diseases. So we want to know what happens when the cells are taking up the rare earth particles. As you can see here, that the particles we are doing these confocal studies, we label the particles with the fluorescent dye, the green, FITC, and we label the red, the lysosomes with red. So what you can see is a lot of yellow showed up, meaning the particles, lanthanides and titanium, are taken up into the lysosome. When look into what happens in the lysosome, we found the lanthanides, the rare earth particles, they seem to change their shape when we do the TEM. Then we zoom into those particles inside the lysosome. What we found is really interesting. The particles change their shape into a needle-like structures. But that does not happen to the titanium. And when we do the EDX, that can tell the elemental composition of these nanoparticles. We found the lanthanides is not lanthanide oxide anymore. They become lanthanide phosphates. But titanium has no changes. So what happens here? We try to understand their mechanism. Then we use a cell-free system that we use a PSF, phagolysosomal simulant fluid, that mimicking the pH levels of the lysosomes, and also containing the phosphates, those chemical components. What we found is that the particle change their shape from spherical gradually into this, grow needles on it. This not only happens to the lanthanides, it also happens to other rare earth oxides. As you can see, they all transformed into that needle-like structure. So to make it a more easily understandable here, we name this sea urchin structures. They do look like sea urchins when everything is ripe. So to study these transformed materials, we do the XRD. Then we found out that they change their composition also from the lanthanum oxides to the lanthanum phosphate. As time goes by, more phosphates are accumulated here. So this is not only a physical transformation, but also a compositional, changes. So to see this, what we think is happening is that the spherical particles, they undergo dissolution. This dissolution process is speed up in the lysosome because they have a low pH. They release the rare earth ions from the particles. So the particle become less, smaller. But the rare earth ions, they bind to the phosphates that's available, in the lysosomal environment to form the rare earth phosphates. These rare earth phosphates, by nature, they like to grow into a needle structure. And the most convenient structure basis is the particle itself. So they grow, they dissolve, they grow, in situ. Then you form these sea urchin structures. So that's what we think is what's happening to these changes. So where is the phosphate from? That's a good question, So then we study, we know these particles will get taken up by the cell, and after a series of transferring into the lysosomes, these are the green are the lysosomes inside of a cell. And we use a fluorescent dye to see whether the lysosome gets damaged, because this sea urchin structures, this transformation, the outcome may be the lysosomal damage. So we use confocal microscopy to stain the lysosomes with a magic red, cathepsin B substrate, You can see the control cells, they have, discrete punctate form of lysosome, meaning they are intact. The cerium or the other transition metal oxide don't induce fluorescent changes. Staining patterns are the same. However, when you look at the rare earth ones, gadolinium, lanthanide, and yttrium, the staining disappears. So that's a sign of lysosomal damage. So then, how these lysosomes are damaged by the rare earth material. We try to, study this. What we see here is what we did for the, TM, for the cell image. You don't see, you see those bubbles. Those are the typical cell deaths induced by these rare earths as showed before. When you zoom in, you see where the sea urchin grows. It's on the inner side of the lysosome membrane. As you can see closely, you have the sea urchin structures on the lysosomal membrane. Also, you see some, the gaps between the lysosomal membrane. Same thing happens to yttrium oxide. They grow the needle-like things on the inner membrane of the lysosome. This does not happen to the other transition metal oxides. Cobalt, manganese, you can see they are intact. They don't grow anything, near the lysosomal membrane. So what happens is what we think. The phosphates is not only from the lysosomal inner solutions, right? But the rare earth is stripping the phosphate from the lysosomal membrane, as I showed here. They grow right there in the lysosomal membrane. So to prove this, we use a cell-free system here again. We synthesize a liposome. The liposome is a lipid bilayer, Then we put some fluorescent dye inside. Then we incubate the liposomes with the rare earths and other transition metal oxide to see what happens. The results are that the rare earth oxides induce the leakage of the liposomes. As you can see, on the left side, the fluorescence leaked out from the liposomes. And when we do the LC mass to determine the molecular weight of the phospholipids, what we found is the phospholipids. So all the phospholipids is lacking 80 Dalton of molecular weight. That's exactly the same weight as the phosphate groups. This does not happen to the the other transition metal oxide, or even the cerium. Cerium is a rare earth oxide, but it's highly indissoluble in the solution. They don't transform. They don't induce this kind of swelling and cell death. So to confirm this, we use the red blood cell hemolysis assay. Same thing. We see whether in the presence of the rare earth oxides, whether they can induce the hemolysis. But to do this, we incubate the red blood cells in a phosphate freeze. Saline. The saline is known, it's a pH 5.5. And you can see in the saline without a phosphate, these rare earths induce hemolysis, except the cerium. But when you have phosphate in the solution, this hemolysis has been inhibited. So that proves that the rare earth transformation, the chemical composition change, the rare earth is grabbing the phosphate from the phospholipids of those lysosomes or the cells. So not only they grab the phosphate group from the lipids, they also grab it from the enzymes, lysosomal enzymes. Here I showed the beta-galactosidase. So this is a major lysosomal enzyme. Then you see when we stain the protein for phosphorylated protein, that the lanthanides has no staining, meaning this phosphorylation has been inhibited. And when we do the enzymatic assay, the lanthanides induce a reduced enzymatic activity, showing that the protein function has been lost. So that's the stripping of the phosphate groups is causing a dysfunction of the lysosomal enzymes. So to just strapialize this means that the lysosomal function has been compromised also. So the lysosomal damage will release all the lysosomal content, and that triggers a pathway that recently really hot, inflammatory pathway. It's called the inflammasome pathway. These inflammasomes play a major role in many, acute and chronic inflammatory diseases like heart disease, lung asbestosis, the brain Alzheimer's disease, right? So the lysosome damage can trigger these inflammasomes. So what I showed here is the NAP3 inflammasome pathway. That it's a major complex that triggers, caspase-1 activation, and then trigger the production of a cytokine called IL-1 beta. So as you can see here, among the many particles, we see that the rare earths induce the activation of the NAP3 inflammasome and the activation of caspase-1. So the caspase-1 is responsible for processing the pro-IL-1 beta into the IL-1 beta. The mature IL-1 beta is a major, inflammatory cytokine. When you have a cold or infection, you have fever, right? So the IL-1 beta is a cytokine that generates fever, the pyogenic. So when we do the screening for all the, metal oxides, you can see that the rare earth ones triggers the highest level of IL-1 beta production. And IL-1 beta production, and the caspase-1 activation, recently people found that they can induce a GSDMD, gasdermin D dependent cell death mechanism called pyroptosis. So this was only discovered, years ago. It's a new type of cell death that induces cell swelling and bubbling up as we saw for the, rare earth materials. And for rare earth material, what I think happens is they go into the lysosome, transform their, their structures, and stripping the phosphate from the lipid bilayer, induce the lysosomal damage, trigger the activation of the NAP3 inflammasome. Then there are two ways of, outcome. The caspase-1 activation produce IL-1 beta, and also cleavage of the gasdermin D. The N-terminal of the gasdermin D, the cleavage, after cleavage, can move to the plasma membrane, and where they form a pore that leading to the leakage of the membrane, the cell swells and, cell membrane ruptures. So to prove that the gas thermin D is forming pores, the major mechanism, we use siRNA to knock down the gas thermin D expression in the Kupffer cells. What we found is you see the red bar. They inhibit the LDH, the large protein release induced by rare earth particles. However, using a scrambled siRNA, they don't have this effect. This is the gas D dependent pyroptosis, induced by these rare earth materials. So does this only happen to the Kupffer cells? We also tried other macrophages like J774A1 and the RAW 267.4.7 and the primary marrow-derived macrophages. You see errors induced, same kind of cell death as the Kupffer cells. And also they induce high level of L1 beta production. And morphologically, you can see they induce in all these cell types, the cell bubbling up and, welling, a typical pyogenic, pyroptosis, in cells. So these are the, for several years we figure out the whole, mechanism of toxicity I mentioned. The cells are taken up, they transform, leading to the stripping of phosphate from the liposome, lysosome membrane, then release the components, trigger NAP3 inflammasome activation, the L1 beta production, the cleavage of the GSDMD, gas thermin D, to form a pore to induce, the pyogenic processes. So these are the, the pathways. It's what we think is happening here. So the L1 beta is the production triggered by these rare earth materials. What's the result of this L1 beta production? That we have found that in the lung, at least, these L1 beta production by macrophages will trigger a series of cellular events by the epithelial cells fibroblasts, leading to the lung fibrosis, the collagen deposition, and also triggers the production of TGF-β, PDGF-AA that play major roles in this lung fibrosis. And these are the, in vivo, can be demonstrated in vivo in the lung. So here I showed the results of the lung fibrosis study. As you can see that the, the miniosol, that a quartz as a positive control induces this production of the blue, collagen deposition in the lung. The gadolinium, erbium, and the, ytterbium oxides, they all trigger this extensive, production of collagen in the lung, showing that they generate lung fibrosis. So that's the eventual adverse outcome that we can prove in vivo. So having known this, what does it help, for us? So how do we make this nanomaterial safer? So as we understand the phosphate binding, from the sea urchin structures are important for toxicity. So then we use a safer design approach by incubating the particles with the phosphate, first. Then we expose to the phagolysosomal simulated fluid. What we see here that the pre-coating prevents the sea urchin structure formation. And they have less phosphate, less than phosphate finding by the XRD. When we test the hemolysis, the phosphate-coated ones do not induce. You see the green, the red stain here, the hemolysis. They don't happen anymore. So the coating, pre-coating, prohibited the hemolysis generated by REOs. So they also do not generate the lysosomal damage and the IL-1 beta production. So we prevented this. So the phosphate-coated particle now is a safer particles compared to the pristine particles. When we do the in vivo lung fibrosis study, you see the pre-coated particle do not generate lung fibrosis like the pristine non-coated. This now is officially a safer particle because we prevented the lung fibrosis. As I mentioned, the ARP conversion particle contains the rare earths that people use for bioimaging. What we show here, we pre-coat the particles with phosphonate, EDTMP, which contains 4 phosphate groups that has a higher binding ability with the rare earth elements. That prevents not only the transformation, but also inhibits the emission, the bioimaging property loss from the transformation process by the rare earths. Also, they do not induce lung fibrosis. So now we have a functional, safer bioimaging agent that can be safely used. So through the similar pathways, NAP3 pathways, we studied many carbon-based materials like carbon nanotube, single wall, multi-wall, different charges, graphene oxides. We found that they trigger the similar pathways like the lysosomal damage, NAP3 inflammasome activation, IL-1 beta production, and the lung fibrosis. So that goes the similar pathways. How do we use this information to have safer materials? Here we use a pluronic, as we discussed, at the Matrix this morning, that pluronic coated the carbon nanotube surface, so they do not induce the following events, like the lysosomal damage. They do not trigger lysosomal damage anymore. They do not induce IL-1 beta production. Also, the lung fibrosis. So that's a safer design for carbon nanotube. So here I'm showing the fumed silica results. They also trigger NAP3 inflammasome pathway, but through a different mechanism. The fumed silica has a lot of the hydroxyl groups on the particle surface. They interact with the plasma membrane lipid bilayer and induce membrane perturbation. That induce the potassium release, from the inner, cells, trigger NAP3 inflammasome activation and induce lung fibrosis. So to make this safer, we do this called titanium or aluminum doping to decrease the surface hydroxyl groups. And the membrane binding ability, so they do not induce membrane perturbation. They don't trigger the potassium efflux and the NAP3 inflammasome activation, and they end up to be safe to the lung. So they do not trigger lung fibrosis anymore. So similarly, we use, a aluminum oxyhydroxide, the nanorods. They trigger the same mechanism, like L1 beta. But here we use a different purpose. We use this to develop a adjuvant effect. That the L1 beta production is not triggering the fibrosis outcome, but they serve as a signal to trigger the, adaptive immunity that we can show that they can be a better vaccine, to replace those alum, the aluminum, salts as the adjuvant. So recently we studied the nanocellulose using the libraries prepared by Phil, and we show the similar mechanism. They trigger the lysosomal damage when cell uptake them, and they trigger L1 beta. So as I show on the right side, we knock out the NAP3 and ASC, that the inflammasome components. Then they stop to produce L1 beta. That shows the NAP3 inflammasome pathway is involved. And when we use this for an adjuvant purpose, that they can stimulate the OVA-specific IgG production, but not the IgE production. So this shows nanocellulose may be a good, adjuvant that can be used in a vaccine. So over the years, we studied probably over 100 different nanomaterials. We can characterize them into different groups. So transition metal oxide, that talked before, that they induce the oxidative stress, induce acute inflammation in the lung. The silica, on the other hand, carbon nanotubes, graphene oxides, or the high ice-phase ratio materials, they trigger as well as the rare earth materials. They trigger NAP3 inflammasome pathway. That long-term, can trigger lung fibrosis, a chronic effect. The silica, the copper, zinc, silver, they can induce lung acute toxicity or chronic toxicity through the releasing of their ions through a dissolution pathway, So that's the summary of our studies over the years. So that's the end of my talk. So thank you very much for your attention.